No, a teraz? Dobrze, przepraszam. Dobrze. No to tak, powtórzę jeszcze raz. Ja nazywam się Marta Aksamit i mam zaszczyt pełnić rolę koordynatora sesji Basic Science, Biotechnology and Biomedical Engineering i to już drugiej części. Serdecznie chciałabym powitać szanowną komisję zarówno stacjonarnie, jak i zdalnie tutaj z nami. Chciałabym przywitać panią lekarz Barbarę Lipkę. Dzień dobry. Oraz pana lekarza Macieja Nowaka oraz pana lekarza Piotra Lewandowskiego, którzy są dzisiaj z nami zdalnie. Szanowna komisjo, pragnę uprzejmie Aha, jeszcze jedna rzecz. Chciałabym wyjaśnić zasady obowiązujące podczas sesji. Kolejność prezentacji odpowiada kolejności zamieszczonej w książce abstraktów. Maksymalny czas na prezentację to 7 minut oraz 3 minuty na dyskusję. Jeżeli prezenter przekroczy czas, zobowiązany jest przejść do wniosków i zakończyć prezentację jak najszybciej. Szanowna komisjo, pragnę uprzejmie przypomnieć, że w przypadku, gdy są Państwo opiekunem naukowym prezentowanej pracy, nie powinni Państwo jej oceniać. W takim przypadku proszę skreślić numer pracy na karcie oceny. Pozostali członkowie tej samej kliniki mogą oceniać daną prezentację. Oceny będą weryfikowane. Wszystkie prezentacje oraz dyskusje muszą być prowadzone w języku angielskim. I tutaj tak, jeżeli decydujecie się, chodzi tutaj o to, że jeżeli decydujecie się na prezentację w języku angielskim, to odpowiadać na pytania również musicie w języku angielskim. Natomiast w tym roku języka nie oceniamy, więc jeżeli zdecydujecie się na prowadzenie po polsku, no to będzie to też potem język polski, więc macie tutaj dowolność. Dobrze. Uprzejmie prosimy komisję o ocenę według następujących kryteriów. Wartość naukowa, punkty od 1 do 5, sposób prezentacji, punkty od 1 do 5, dyskusja od 1 do 5 i wkład własny od 1 do 5. Po zakończeniu sesji prosimy o zwrot kart ocen do koordynatora sesji, czy też mojej asystentki dzisiaj, lub odesłanie wypełnionych kart ocen drogą mailową na ten sam adres, z którego zostały one wysłane. I myślę, że możemy już zacząć, także zapraszam pierwszą osobę do prezentowania. A jest ona chyba z nami online. Proszę powiedzieć, czy jest tutaj z nami pierwsza prezentująca osoba?
Yes, I'm here.
Super, świetnie. No to oddaję głos. Proszę udostępnić też ekran z prezentacją. No i jak tylko będziesz gotowa, to proszę zaczynać.
Okay, so I would like to start. Good morning, everybody. I am Hanna Krawzik. I am member of Scientific Research Student Club at Laboratory of Metabolomics and Proteomics in Clinical Research Center, Medical University of Białystok. I have the honor to present you work titled Histopathological Characterization of PAX2 Syndrome, a first look into the tissue structure. My tutor of the work is MSc Arkadiusz Żbikowski. So, starting our presentation, I would like to say a few things about PAX2 syndrome. What is PAX2 syndrome? It's an ultra-rare genetic disorder caused by de novo mutation. And this important to say there are around 100 diagnosed patients worldwide, and 6 of them lives in Poland. This picture show MRI taken performed on the patient diagnosed with the syndrome and come from Stoyan-Lisher review. So using Stoyan-Lisher review, we are able to collect the described cases, 29 cases, and all of them shows epilepsy. This is usually the first symptoms that start the diagnosis of the disease. Another neurological symptoms are neurodevelopmental and motor delay, speech delay, hypotonia, behavioral disorders, and other are like you can see on this slide. Other symptoms, other than neurological, are limb distal malformation, ocular abnormalities, dysfunction of urogenital system, hematological disturbances, and cardiac septal defects. So, knowing the symptoms of the syndrome, we can compose aim of our study to investigate potential changes within structure of lungs, kidneys, and liver in PAX2 syndrome. So I would like to show you methodology and study design of our research. We took 12 adult mice. They have 10 to 12 weeks old. 6 of them have knock-in PAX mutation and 6 of them are wild type. As I said earlier, we collect tissue from the kidney, lung, and liver and perform histology staining. H+E for anatomical structure, vancos for calcification, and trichome for collagen deposition. We also use qRPCR, compare, compose, obtain remodeling tissue genes structures, remodeling gen tissue. They are collagen, MMPs, TIMPs, vimentin, and alpha SMA. So I would like to show you our results. We start from the kidneys. In the H+E stain, you can see and I can see any significant change between control and PAX2, same as in the Vonkos staining, and same as in the trichome staining. As I said earlier, we also do qRPCR, but we don't observe any significant change like increasing, decreasing level of these genes in the kidneys. Another tissue is lung. In the H+E staining, we don't observe any significant change between control and PAX2. Same as on the Vonkos and same as in the trichome. But you can see we observe change between MMP25, collagen 3A1, TIMP1 and TIMP4. And another structure is liver. In the H+E staining, we don't see change between control and PAX2, same as on the Vonkos staining. But in the trichome staining, we can observe change. You can see on the PAX2 is different than control. And you can see on this yellow square, this dark blue line suggests collagen deposition. Because as I say, trichome is the collagen deposition. So we can think about process in the liver that we see also in the qRPCR. We see MMP9 increasing level, MMP12, collagen A1A, collagen 3A1, TIMP1 and TIMP3, and vimentin. So what is the conclusion of our work? This finds suggest that PAX2 syndrome may contribute to dysregulation of tissue remodeling process. Our mouse model represents young adults, and all observations were spontaneous. Therefore, it is plausible that tissue remodeling process may be more advanced in older individuals. And these results highlight the need to expand our research. So I would like to thank you for your attention, and also I would like to thank all the people who are involved in this scientific research.
Dobrze, dziękujemy bardzo za tą prezentację. Jeszcze tylko krótka informacja do tutaj komisji oceniającej, ponieważ widzimy, że jest to zapisane jako temat numer 2, dlatego proszę o uwzględnienie tego, że to był temat numer 2, bo widocznie osoba, która miała temat numer 1 się albo z nami nie połączyła, albo miała jakieś problemy. Dlatego jeszcze raz powtórzę tutaj zarówno stacjonarnie, jak i zdalnie, że ta praca widnieje pod numerem 2. Więc proszę to uwzględnić w ocenianiu. No i teraz myślę, że można przejść jeszcze do dyskusji.
Thank you for your presentation. I have 2 questions. Firstly, what was your contribution in this work, and who did the statistical analysis? Did you do it yourself, or you send it to some statistical people who do the statistics?
Yes, I happy to see you to hear your question. My part of the research was analyze photo of the tissue, preparing PCRs, but unlucky I can't work with animals now. So in the future, I would be happy to get my license to work with animals. And speaking about PCRs and analysis of the PCR, I do part of the work, but all the analysis made my tutor, Arkadiusz Bikowski, and Scientific Students Club part of Center of Regenerative Medicine, Krystian Czołpiński.
Okay, thank you for your answers. Personally, in my opinion, we should do the best in our part and put our best contribution. We don't have to do the statistical data. So that's why I ask, because I think doctors and biologists are not there to do most of the statistical data. We should divide our work and put our best impact in the field which we are good at.
Yes, this is true.
Okay, and another question, because you found higher collagen deposition in the liver. How do you, maybe this is a question from my interest. Do you think, how does it contribute to the symptoms of this disease? Do you find any collaboration between that? Because that was the only place where the changes were present, but the main symptoms are seizures. And I would also like to ask, why didn't you investigate the brain?
Yes, we also took from our mice brain samples, and my tutor presents yesterday work about proteomics levels. And you can see on these pictures, we see that B-glycan is mostly responsible for collagen assemble, and in the liver is increasing level of them. But in the brain, we also see any changes but we don't put it in our work because we are focused on liver, lung, and kidney now. But we have the data, and we plan to publish our work later.
Okay, thank you. I think that would be really interesting to include.
May I ask also the question?
Yes.
Thank you very much. I want to a bit disagree with my colleague. I think that as a principal investigator, you have to know about all aspects of the work, including statistics, but not about that I want to ask. I want to ask of PCR. Did you follow any guidelines for providing quality in PCR statistic? For example, Mikey guidelines did you follow? And which housekeeping genes did you use as a control?
Okay, this is very interesting question. Thank you. I taking part to prepare our samples to the qRPCR, but I don't take part on the statistic levels of the statistic work that I only to see. But I don't know, but I would love and like to ask my colleagues, as I say earlier, that do most of the work using PCRs.
Okay, thank you very much.
Thank you.
Dobrze, czy są jeszcze jakieś pytania? Dobrze, to jeśli nie ma, to ja tutaj bardzo dziękuję za tą prezentację. I chciałabym teraz zapytać, czy jest może z nami autor pracy numer 1, czyli czy jest z nami Łukasz?
Tak, hello everyone. Yes, I am.
Super, dobra. No to w takim razie w tym momencie wrócimy się do pracy numer 1. Jeżeli możesz, to proszę o udostępnienie ekranu i kamerki siebie. I teraz jeszcze raz informacja dla oceniających. Będzie to teraz praca numer 1, później już pójdziemy w normalnym trybie.
Good morning, everyone.
I jeszcze jakbym mogła tylko, właśnie na pokaz slajdów, żeby był pełen ekran.
Yes, of course. Okay, let's start. Good morning, everyone. My name is Łukasz Cięciała, and I am a member of Scientific Lab of System Engineering and Biology of the Biotechnology Center of the Silesia University of Technology. And also, I am a member of FEPS Young Poland. Today, I have a pleasure of presenting a research project concluded in collaboration with PhD Dr. Małgorzata Damiec-Organiściak and also Professor Magdalena Skonieczna. The aim of the project was to investigate the possibility of inducing programmed cell death, specifically ferroptosis, through genetic modification using CRISPR-Cas9 technology. This project aimed to deepen our understanding of the mechanism for the cell death. Programmed cell death and its subtype regulated cell death are mechanisms by which the body eliminates abnormal or damaged cells. In recent years, increasing attention has been paid to ferroptosis of RCD that differ mechanically from the better known apoptosis. Ferroptosis depends on the presence of iron and reactive oxygen species, which cause irreversibly damage to the cell membranes through lipid peroxidation. A key component in protecting against ferroptosis is the enzyme GPX4, which detoxifies lipid hydroperoxidase. In our project, we use CRISPR-Cas9 double nuclease technology to knock out the GPX4 gene and assess the effect of this modification in cells treated with Arrestin. Arrestin is, as you know, ferroptosis inducer. And also on the slide, I shows a simplified model of the ferroptosis pathway. Iron enters the cells via cells, iron can be reduced and participates in the Fenton reaction, leading to the formation of ROS. And excess of ROS causes lipid peroxidation in cell membranes. GPX4 contracts this process by detoxifying lipid peroxidase. When GPX4 is knocked out, the cells loses the protection and become sustainable to ferroptotic death. Other key proteins include ACSL4 involved in lipid metabolism, as well as protein 2 and ferroportin, both associated with iron metabolism. In my experiment, we used two Hackett cell lines, one wild type and one genetically modified using CRISPR-Cas9. Ferroptosis was induced using Arrestin, which inhibits the cysteine transporter, leading to reduced glutathione level and increased ROS level. Cell variability was associated using the MTS assay. Gene expression was measured with real-time qPCR. Protein level are analyzed via Western blot, and the ROS and glutathione level were evaluated using fluorescence and spectrophotometric methods. Effect of genetic modification and cell viability. This graph shows GPX4 mRNA level in Y type and KO cells after 24 hours treatment with our inducer ferroptosis Arrestin in 2 doses, 5 and 10 micromole. In the KO9 GPX4 transcript level was significantly lower, confirming the effectiveness of the knockout. Additionally, the results suggest the Arrestin may further reduce GPX4, expression in Y-type cells and changing its prophylactic effects. Mitochondrial activity is also viability. Was associated with using the MTS assay. The results showed a clear decrease in viability in GPX4KO cells compared to wild type, especially after treatment with 10 micromole of Arastin. This suggests that the absence of GPX4 leads to faster loss of metabolic function and cell death in response to oxidative stress. The results showed a significant increase in reactive oxygen species in GPX4 cells. At the same time, total glutathione level decreased after Arastin treatment, inducing a weakened capacity of detoxify ROS. The combination of high ROS and low GSA is a hallmark of ferroptosis. Lipid pathway SEL4. An increase this gene expression in the KO line after Arastin treatment confirms activation of the lipid peroxidation pathway. ACL4 participates in the production of long-chain fatty acids, which are prone to peroxidation. Its underregulation correlates with increased cell susceptibility to ferroptosis. Iron metabolism pathway. We examined three genes. We measure the level of TFRC. We see in cells Y type, we don't see big decrease of level this gene, but we have huge increase in cells knockout. Level of ferroprotein, we have increase in which we see increase with every doses 5 and 10 micromole and in our Y-type cell line. But also we see don't change or very little change level of mRNA level of this gene in KO cells. Level of promin 2, we have increase in cell Y type, and we don't have this expression in our knockouted cell line. Summary, CRISPR-Cas9 as a research tool. The technology enabled to effective silencing of the GPX4 gene, confirming its usefulness in modeling cell death. The absence of GPX4 made the cells more susceptible Arastin-induced ferroptosis. Highlight the role of the enzyme is antioxidant protection. Increased expression of ACLS4 and decreased expression PROM2 indicate a strong link between ferroptosis metabolism, lipid metabolism, and iron homeostasis. Thank you so much for attention. If you have any question, I answer.
Dobrze, dziękujemy bardzo za tą prezentację i teraz zachęcam również do dyskusji i zadawania pytań.
Thank you very much for this nice presentation. Could you please tell your contribute to the work, which part you have performed?
Yeah, sure. I do knockout this time like 3 months, and also I do most of PCR. My tutor do also a PCR, but I the most focus on the cell culture and PCR. and my supervisor do the rest stuff.
Okay, thank you very much.
Czy są jeszcze jakieś pytania? Dobrze, myślę, że w takim razie nie ma już więcej pytań, więc dziękuję bardzo za tą prezentację i dyskusję.
Thank you.
I poprosiłabym teraz o pracę numer 3. Ona również powinna być chyba zdalnie. Czy jest z nami może Alicja?
Tak, jestem. Dzień dobry.
Dzień dobry, super. W takim razie proszę o udostępnienie ekranu i właśnie kamerki. Super, już to widzę. Teraz jeszcze tylko udostępnienie ekranu z prezentacją. No i oczywiście, jeżeli tylko będzie pełna gotowość, no to można zaczynać.
Okay, good morning. I would like to walk you through the presentation about the lung resident fibroblasts in airway remodeling across experimental asthma phenotypes. I would like to say about the asthma. What exactly is asthma? Asthma is an inflammatory airway disorder in which inflammation plays a crucial role. And asthma has different phenotypes. And that's why asthma is really heterogeneous. And in asthma, inflammation and airway remodeling is associated with the processes underlying this disorder. And the most important components are lung resident fibroblasts, which are responsible for maintaining tissue homeostasis. However, during the inflammation and remodeling, they producing collagens to the extracellular matrix. And that's the problem. It's really challenging in treating asthma. And we would like to know more about the role during these processes. So that's why our aim was to investigate the role of these lung fibroblasts in airway remodeling and fibrosis across different inflammatory asthma phenotypes. And to do that, we used the different models, mice models, and we perform complementary in vitro approaches. And the first model was acute T2-low type airway inflammation asthma, in which the mice were stimulated with house dust mice HDM extract in 10 or 100 micrograms for 2 weeks. And the second one was chronic non-2 type airway inflammation asthma, known as neutrophilic, in which mice were stimulated with HDM extract extended to 12 weeks in 100 micrograms. So to uncover the molecular features of these phenotypes, we perform a transcriptomic characterization of the lungs. And we can see the differences, mRNA signatures across these phenotypes. However, they also reflect similarities. And we also observed the top differentially regulated pathways that were commonly upregulated in all of these 3 models. However, they were implicated in inflammation rather than in airway remodeling. So that's why we would like to we did curated analysis and we were focused on the fibroblast growth factors and myofibroblast differentiation markers, which are really important in these processes. So we can see that FGF23 was mostly upregulated within these three models. However, for example, FGF2 was downregulated commonly. In myofibroblast markers, we can see the TGF-BR1 was commonly upregulated. And we can see here we measured the expression of TGF-beta1, which is the orchestrator of these processes during the fibroblast to myofibroblast differentiation. And we can see the increasing the gene expression of the TGF-beta in all of these models, while in bulk fluid, we can see that in acute model of HDM 100 and chronic model was upregulated level, and HDM 10 in acute model was a trend. Surprisingly, ACTA2 gene was, there were no differences observed. However, in a chronic model was lower expression. We can see also alpha SMA here, and in all of the model was lower expression of this one. We also measured the number of fibroblasts in the lungs. And in acute models, the number was significantly upregulated together with FGF2 in these acute models. We also can see on these pictures, we measured the presence and the role of fibroblasts, and we can see in these lungs the alpha SMA in subepithelium. The thickness and the volume was observed upregulated in acute HDM2 and chronic model. And also we measured the FGF21 and the level was increased in acute models. That could mean that maybe the organism, because of the property of this FGF21, like this is antifibrotic, so maybe the organism was efficient to fight with fibrosis or other processes underlying remodeling. Yes, so that's why maybe a thing. And also we did in vitro approaches. We used the primary human lung fibroblasts. I stimulated them with several components, such as TGF-beta1, FGF2, FGF21, and the mix of these components. And I did the immunofluorescence staining, for example, and I will present you the pictures. Other samples were analyzed using different analyses. And we can see here the visualization of tunnel assay. And these cells are from patients with asthma. And they were stimulated, and we can see the control stimulated with the components, and the red dots especially, which indicate the apoptotic areas. And the second one was the cells from the healthy individual, and we cannot see here any differences like before. So I would like to say that these are in vitro approaches are preliminary data, really fresh. I did it as I am almost master students. So yeah, I would like to share with you my data, and I would like to highlight that these cells were really tricky, and we were shocked because they were detached from the surfaces, and it was not really good for cell culturing. So the closing thoughts are that the inflammatory asthma phenotypes elicit distinct fibroblast responses, and the fibroblast growth factors regulate these processes. And extracellular matrix deposition is really important here. And that's the open question. Maybe we can think about that. Does maybe the airway remodeling environment shape the fibroblasts of myofibroblasts within this environment, specific airway remodeling environment, or maybe is something deeper, and we can go there. Thank you so much.
Dziękujemy bardzo za tą prezentację i teraz przechodzimy do momentu zadawania pytań i dyskusji.
Thank you for this interesting research, and I suppose your contribution was huge. I would like to know, how does the finding correspond with existing literature on this topic? Because I assume you did a research on this topic, and how does your finding correspond with, or is there even a wide literature of this topic?
It's a funny story because I sent an email to Boris Hinz, for example, who is known in fibroblast cell culturing. So I would like to know more about the cell culturing, especially these fibroblasts. And I found a lot of informations and additional information in email from him. He sent me also a chapter from his book, and that's why I chose this concentration of the components like TGF-beta stimulation of the fibroblasts, FBS changing during the cell culturing and passaging. So that was the core. As well as he provided me with the pictures from his articles, from his book, and it was my basement.
Thank you.
Okay, can I go deeper for the question that the colleague asked? Did you search for the literature using, for example, PubMed or Google Scholar for the impact of TGF-beta on fibroblasts? Because the topic is rather well known that TGF-beta promotes tissue remodeling and acting and changes in fibroblasts. So how your study is different than those studies done before?
My study is different because of the fact that we used as well as mice models with different phenotypes and human cells, and it's really interesting. As I said, it's preliminary research. I would like to go deeper and check the environment which is the best for these cells. But I would like to highlight the most important thing is that I would like to go deeper. For example, when I chose these concentrations of TGF-beta, for example, and I mixed these components, I would like to see whether these cells will behave in another way, when we have fibroblast to fibroblast differentiation, how this TGF-beta will behave with other components, and And I would like to say that I would like to see the clotho, for example, protein, which is associated with FGF21, which is producing by the liver. And maybe here, as FGF21 as a hormone acting in the organism, we can just at this level treat or maybe somehow manipulate with this to prevent fibroblast to myofibroblast transition in remodeling or fibrosis, to prevent producing scar, because scar in fibrosis is associated with stiffness of the tissue. So that's why the lung is not properly functioning. So this is the problem with asthma, for example, in chronic asthma. And when we have these problems, mostly in chronic asthma, which is related to T2, which is related to neutrophilic type. So these patients with neutrophilic types, so non-T2 dependent or T2 low dependent, are really reluctant to novel biologics treatment and corticosteroids treatment nowadays.
Okay, thank you very much for this very, very deep story. Thank you very much.
Dobrze, w takim razie dziękujemy za te pytania oraz dyskusję. Dziękujemy również za tą prezentację i zapraszamy teraz autorkę pracy numer 4, która powinna być tutaj stacjonarnie z nami. Czy jest może z nami? Super, dobrze, świetnie. To zapraszam, tutaj włączymy.
Good morning, everyone. I am Natalia Kitala, and I'm happy to give you a presentation about the role of minocycline in modulating Th2 cytokine expression after subarachnoid hemorrhage in a rat model. I will start my presentation with the subarachnoid hemorrhage itself. It accounts for around 20% of all strokes, and it is most often caused by a ruptured cerebral aneurysm. Around 20% of survivors live with serious disabilities such as paresis, limb paralysis, or speech and consciousness disorders. This puts a huge emotional burden on patients, and that's why it's so important topic. What makes subarachnoid hemorrhage particularly dangerous is that it triggers a strong inflammatory reaction that drives both early and delayed brain injury. Early brain injury develops within the first 72 hours. When the blood enters the subarachnoid space, it activates microglia and astrocytes, which are brain immune cells. They start to release pro-inflammatory cytokines, which break down the blood-brain barrier, allowing more immune cells to infiltrate the brain and to cause brain edema, oxidative stress, and neuronal death. This early inflammation phase lays a foundation for the delayed brain injury. This state affects up to 40% of the patients, typically between days 4 and 14 post-subarachnoid hemorrhage. Here, chronic microglial activation and inflammation continues and contributes to vasospasm and delayed cerebral ischemia. Microthrombi also forms due to inflammation, further impairing brain perfusion. The delayed brain injury usually occurs as new neurological deficits, and as you can see, inflammation plays a crucial role in both early and delayed brain injury, and that's why it's a key therapeutic target. But inflammation is not one-sided, because the immune system also responds with the anti-inflammatory mechanisms, and a key part of that is the Th2 cytokine groups, mainly interleukin 4, 13, 5, and 10. Interleukin 4 and 13 promote the M2 anti-inflammatory microglial phenotype. This helps to reduce inflammation and helps to reduce brain tissue. Both cytokines also promote production of interleukin 5. And here things get more complex because interleukin 5 is in fact a part of Th2 cytokine group, but it may worsen the outcome by recruiting eosinophils and contributing to brain edema and hydrocephalus. Then we have interleukin 10, which is a strong anti-inflammatory cytokine that reduces inflammation and helps brain repair. However, after a severe brain injury, such subarachnoid hemorrhage, it may become overexpressed into a condition called CARS, which is a compensatory anti-inflammatory response. This state suppresses immunity and exposes patients to infections like pneumonia or sepsis, which are the most common causes of death after subarachnoid hemorrhage. With all that being said, we can move to the aim of our experimental study, which was to assess the effects of minocycline, which is an antibiotic with well-known neuroprotective and anti-inflammatory properties, on the expression of Th2 cytokines and to explore its potential neuroprotective role in a rat model of subarachnoid hemorrhage. So how did we study this? Subarachnoid hemorrhage was induced in rats by a prechiasmatic injection of fresh arterial blood. After the procedures, rat either received minocycline daily or no treatment. Then animals underwent euthanasia on their first, third, and seventh post-subarachnoid hemorrhage, and the lymphocytes were isolated, cultured for 24 hours, and the cytokine levels were measured by a bioplex multiplex immunoassay system. And now I'm happy to share our results with you. As you can see in this slide, all cytokines measured showed a trend toward increased concentration on day 7 post-subarachnoid hemorrhage. Interleukin 10 and 13 showed the most significant increase after subarachnoid hemorrhage, indicating a counter-regulatory anti-inflammatory response. Minocycline altered this profile. In the early phase, it increases levels of anti-inflammatory cytokine, suggesting a neuroprotective properties. In the second phase, it reduces level of interleukin 5, possibly ameliorating the inflammation, and at the same time, it reduces level of interleukin 4 and prevents the late interleukin 10 surge observed in the untreated group. This might suggest impaired regeneration. However, I have mentioned earlier that overexpression of interleukin 10 may suppress the immunity and expose patients to life-threatening infections. So this particular effect of interleukin 10 of minocycline, might be actually beneficial for the patient. To sum everything up, we can say that minocycline may offer a phase-dependent protection after subarachnoid hemorrhage by influencing the Th2 cytokine dynamics, and I truly believe that even small insights into the immunology of subarachnoid hemorrhage may bring us closer to the better outcomes for the patients. That's all from me. Thank you very much for your attention, and I will be happy to answer any questions.
Dobrze, świetnie, dziękujemy bardzo za tą prezentację i tak jak w poprzednich pracach zachęcam teraz do dyskusji lub zadawania pytań.
I would like to ask on a possible clinical implication. On which stage would you recommend to introduce the minocycline to the patient? Because I don't know if I got it or not.
I think that basing on our findings, we can suggest administration of minocycline in the delayed phase of the subarachnoid hemorrhage, but I think that this is too early to state this timing of administration, and the further studies are needed. But it actually affected the cytokines level in the second phase the strongest.
Okay, thank you for your answer.
W takim razie, jeżeli nie widzę żadnych innych pytań, to bardzo dziękuję za tą prezentację i zapraszam następną osobę. Tu też ma być właśnie stacjonarna. I tak samo.
Ja chciałam jeszcze dodać, że zachęcamy do zadawania Państwa z widowni pytań, bo na pewno ciekawi Was praca koleżanek i kolegów. Niektóre aspekty może warto dopytać, jeżeli coś Was zaciekawi.
Na ostatniej części mieliśmy nawet żywe dyskusje.
Tak, tak. Wczoraj uczestniczyliśmy właśnie w sesji chirurgii, gdzie od publiczności zaszło było co najmniej 3 pytania, bo myślę, że Was najbardziej interesują nawzajem swoje prace.
So, good morning. My name is Nadzieja Krawiec, and I'm representing the Department of Physiology at the Medical University of Silesia in Katowice. And I have prepared a presentation entitled CXCL1 in subarachnoid hemorrhage, the effect of minocycline on its activity. Firstly, I would like to say a few words about what subarachnoid hemorrhage is. SAH is a Swiss condition where the blood leaks into the subarachnoid space, and often after a ruptured aneurysm. Even if the patient survives the initial bleeds, there's still a high risk of secondary brain damage due to inflammation. And one of the key molecules involved in the inflammation is the chemokine CXCL1. It is released after tissue damage, and it attracts immune cells, mainly neutrophils, to the brain. On the other hand, we know that minocycline, a tetracycline antibiotic, has been shown to reduce this neutrophil activity and brain inflammation after SAH, and it appears to exert these effects by modulating the expression of pro-inflammatory chemokines, including CXCL1. CXCL1 binds to the CXCL2 receptors on neutrophils and endothelial cells, and it activates a lot of inflammatory pathways that can worsen the brain damage by promoting processes like production of reactive oxygen species, degranulation, or chemotaxis. So based on these findings, we decided to further investigate the effects of SAH and minocycline on CXCL1 production by lymphocytes. To explore this process, we used rodent model of SAH. A total of 250 microliters of arterial blood was injected into the prechiasmatic cisterns. One group was treated with minocycline at a dose of 1 milligram per kilogram daily. And then the blood samples were collected on days 1st, 3rd, and 7th. Lymphocytes were then isolated and cultured for 24 hours, and CXC1 levels were measured in this culture media. And these are the results. After SAH, the CXC1 levels were reduced compared to the SHAM group, and this decrease was continued slightly up to 72 hours. Then by day 7, we observed a strong increase in this chemokine concentration, suggesting a delayed immune activation. The group treated with minocycline had significantly lower CXC1 levels by day 7, suggesting that the drug can suppress the late inflammation. As observed here, we see that early after SAH, the CXC1 levels were reduced due to immune suppression, and minocycline may help regulate and normalize this chemokine concentration between 24 and 72 hours by reducing this excessive immune suppression, but by day 7, its anti-inflammatory effects dominate and CXC1 drops again. These fluctuations over time may be influenced by molecules like MMP7 and ACKR1. MMP7 releases active CXC1 by cleaving it from the cell surface from glycosaminoglycans, and it is boosting the inflammation. ACKR1, on the other hand, binds to the CXC1 and limits its activity by protecting it from reaching the CXCR2 receptors. So that can help regulate the immune cell migration. These findings suggest that minocycline may influence the CXC1 levels after subarachnoid hemorrhage through mechanisms involving molecules like MMP7, ACKR1, and given the potential role of CXC1 in regulating the immune responses after SAH, it is crucial to gain a better understanding of how minocycline modulates the CXC1 levels in a time-dependent manner to potentially reduce the risk of secondary brain damage. And that's all. Thank you for your attention.
Dziękuję bardzo za tą prezentację również i tutaj tak samo zachęcam do zadawania pytań zarówno tutaj przez komisję, jak i przez komisję tutaj online'owo. No i tak jak już wcześniej było wspomniane, również zachęcam publiczność.
I would, of course, like to know about your contribution to the work.
I research articles on CXC1 and the potential role of in subarachnoid hemorrhage and how it can be affected then by minocycline. And based on the data on how CXC1 levels change over time in this study, I created this presentation.
And did you participate in the experiments?
No, unfortunately, no, I couldn't.
Okay, thank you.
Dobrze, to w takim razie, jeżeli nie ma już żadnych pytań, to przechodzimy do pracy numer 6 już. Tak, dobrze. Do pracy numer 6 i ona również powinna tutaj być stacjonarnie i super. Dobrze. Tutaj przewijamy, tutaj. Daj mi na chwilę na prezentację. Okej. Tutaj. No. I tutaj jest to. Ale on tylko klikać będę. Okej, nie powinno być. Tutaj do przodu, do tyłu, tutaj masz pointer.
Good morning, everyone. My name is Lidia Korczyńska, and I am delighted to stand here and present to you our findings. The aim of our study was to determine the levels of interleukin-1 beta, TNF-alpha, MIP-1 alpha, and MIP-3 alpha, cytokines in circulating lymphocytes following subarachnoid hemorrhage, both early and late post-injury, and assess minocycline's effect on these levels. Subarachnoid hemorrhage is 20% of all strokes, and it's related to sort of cytokine storms and that's why it's a hard to control process. Our work consisted of finding every detail about interleukin-1 alpha, TNF alpha, MIP-1 alpha, and MIP-3 alpha. It was important to determine how they cooperate with each other and how it results in SAH outcomes. These pro-inflammatory cytokines are secreted by microglia, astrocytes, and damaged endothelium. After early injury, MIP-1 alpha and MIP-3 alpha levels rise rapidly with vasoconstriction, triggering microglial and endothelial activation, which promotes interleukin-1 beta and TNF-alpha release. In turn, they further upregulate MIP-1 alpha and MIP-3 alpha. As we can see on this diagram, creating a positive feedback loop that amplifies inflammation, drives neural injury and edema, and contributes to poorer outcomes of SAH. The study was conducted on 120 male Wistar rats, weighing 250 to 300 grams. In both the SAH and SHAM groups, a polyvinyl catheter was inserted into the cisterna magna above the optic chiasm. Subarachnoid hemorrhage was induced in SAH group by injecting 250 microliters of arterial blood from a donor rat via the catheter. Selected SAH animals received intraperitoneal minocycline at 1 milligram per kilogram in 1 milliliter of saline. Rats were euthanized at 24 hours, 72 hours, or 7 days post-injury. Following anesthesia, venous blood were collected, lymphocytes were isolated and cultured for 24 hours. Cytokine levels were measured using a bioplex multiplex immunoassay system. We observed time-dependent changes in cytokine levels following subarachnoid hemorrhage. Interleukin-1 beta initially decreased, but by day 7, it showed a marked increase, reaching approximately 12 picograms per milliliters. A similar trend was seen for MIP-1 alpha, which peaked about 450 picograms per milliliters on day 7. TNF alpha reached its maximum at 24 hours, about 55 picograms per milliliters, and remained relatively stable afterward. MIP-3 alpha showed the highest levels at 24 hours. It's about 0.9 picograms per milliliters and declined significantly. Among all measured cytokines, only interleukin-1 beta demonstrated a statistically significant decrease on day 7 in animals treated with minocycline. Minocycline helps reduce brain edema by modulating the neuroinflammatory response. It lowers interleukin-1 beta levels through a dual inhibitory mechanism. By suppressing NF-κB activity, it limits the synthesis of pro-interleukin-1 beta, and by inhibiting NLRP3 inflammasome activation, it reduces the release of active interleukin-1 beta. This likely explains why minocycline has a stronger effect on interleukin-1 beta than on TNF-alpha, which is secreted directly in its active form and doesn't depend on inflammasome activation. To sum up our presentation, minocycline inhibits microglial activation, leading to lower levels of multiple pro-inflammatory cytokines. Its antioxidant and anti-apoptotic properties further contribute to neuroprotection. Thank you for your attention. And before the discussion, I'd like to invite the second author, Julią Kazalska.
Dziękuję bardzo za tą prezentację i zachęcam tutaj do zadawania pytań i udziału w dyskusji.
Always, I have to ask about your contribution. Did you participate in any part of the laboratory work or watch it?
Our main work was to search all the articles about the cytokines because we chose the cytokines from the list of cytokines that are injected, like maybe not injected, but that are noticed in subarachnoidal hemorrhage, and we find all the articles, how they level rises and how they work in subarachnoidal hemorrhage, and we compared it with the results of our tutor, Dr. Gendosh De Carillo, and we made this presentation.
Thank you.
Thank you.
No to teraz zapraszam na kolejną prezentację, już 7. Dobrze. No to... Jest wskaźnik. Tutaj czerwony jest wskaźnik. Tylko nie wiem, czy jak teraz przełączę. Tutaj się przełącza. Tutaj do przodu, a tutaj teraz sobie tylko sprawdzę jeszcze.
Nie, myślę na spokojnie. Już mogę zaczynać, tak? Okay, so welcome everyone. My name is Karolina Jankowska. I am a third-year medical student at Silesia Medical University here in Katowice, and I'm representing the Student Scientific Association of Clinical Genetics. I'm very honored to be here and have a chance to be presenting to you a project which title is Role of Splicing Regulators ESRP1 and ESRP2 in epithelial mesenchymal transition in bladder cancer cells. So the presentation outline, firstly, I will talk about bladder cancer. Then I will get you more familiar with epithelial to mesenchymal transition. Next, I will describe our recent study results and conclusions which we have drawn from this part of the project. So starting with bladder cancer. Bladder cancer is the 9th most commonly diagnosed cancer worldwide. Its incidence is 3 times higher in men, especially after the age of 50, than in women. Higher incidence has been documented in Southern and Western Europe as well as in Northern America. About 90% of patients are suffering from urothelial cancer, also called the transitional cell carcinoma. This is the most common subtype of bladder cancer, which is developing from the transitional cells, which are lining the urinary tract, including the bladder, the ureters, and the renal pelvis. The metastatic progression of bladder cancer is still insufficiently studied, highlighting the need for further research on the molecular mechanism underlying this cancer. So the main mechanism which is responsible for the acquisition of migratory capacity in cancer cells with epithelial origin is epithelial to mesenchymal transition. In abbreviation, it's called EMT. During this process, epithelial cells, which are characterized by the front-to-back polarity, tight intercellular connections, and low migratory capacity, they change their morphology into mesenchymal cells, which are characterized by their high ability to metastasize. The cause of the described changes can be attributed to various biological factors like hypoxia, cytokines, or growth factors, the largest of which is TGF-beta. Consequently, this will result in a synthesis of such transcription factors like snail, SLAC, twist, ZEP1, or ZEP2, which are responsible for the expression of specific genes in the cells. Consequently, this whole process will lead to a higher expression of mesenchymal markers, for example, vimentin, and simultaneously there will be a decrease in epithelial markers, for instance, E-cathedrin. EMT is a process which is regulated by post-transcriptional mechanisms, among others, ESRP1 and ESRP2. The previous studies have documented the role of ESRP1 in breast, lung, and pancreatic cancer. However, there are no reports yet on the role in the EMT process in bladder cancer cells. So the aim of the study is to investigate whether splicing regulators ESRP1 and ESRP2 are present and involved in the EMT process in bladder cancer cells. So moving on to the next point. In our study, we looked for EMT profile in 4 commercially available cell lines, 2 from non-invasive, and RT4 and 5637, and 2 from invasive, T24 and UMUC3. The morphological evaluation showed that the cell lines from non-invasive bladder cancer have a phenotype more characteristic for epithelial cells, especially RT4. On the other hand, 2 invasive bladder cancer lines have a phenotype more typical for mesenchymal cells, as you can see here on these 2 pictures. And it was also, this is a picture from a light microscope. To confirm differences on a phenotype level, we analyzed the expression levels of 4 EMT markers, which are ESRP1, ESRP2, E-cathedrin, and vimentin. Firstly, we did it on an RNA level, which you can see, and our outcomes are suggesting us there's a very high expression of ESRP1, ESRP2, and E-cathedrin in RT4. To remind you all, this was our non-invasive line. And at the same time, there's a very high expression of vimentin in UMUC3 line, so the invasive line. The results obtained were confirmed at the protein level with the use of Western blot analysis. And as you may see, we also confirmed it on a protein level in comparison to the RNA level. In the next part, we checked which transcription factor plays a key role as an inducer of the EMT process. So using RT-qPCR method, we analyzed the expression levels of 4 transcription factors, ZEP1, SNAI, SLAC, and twist. As you may see, our outcomes has given that there's a very high expression of ZEP1 in UMUC3 line, which may indicate that this is the key inducer of the EMT process. I'm sorry. In the next step, we made a transient knockdown of ESRP1 and ESRP2 in two non-invasive lines, which are 5637 and RT4 using siRNA. So starting with the top line, as you may see, here we have SIESRP1, and this transient knockdown has an impact on the expression of these four proteins. So after this transient knockdown in both lines, you may see that there's a lower expression level of ESRP1 and ecathedrine, and simultaneously there is an increased level of vimentin and ZEP1. The same situation goes for SIESRP2. As you may see, that after this transient knockdown, the expression levels of ESRP2 and ecathedrine one more time in both lines, they have decreased, and at the same time, the levels of VIM and ZEP1 has increased significantly. What we did next is a stable overexpression of ESRP1 and ESRP2 in the UMUC3 line, so our invasive line. So in the top line, you may see the transfection efficacy in comparison to the control gene, which was GFP, and our outcomes are really satisfactory. In the bottom line, you may see how this stable overexpression has an impact on the expression of EMT markers, which are ecathedrine, VIM, and ZEP1. And as you may see, after both ESRP1 and ESRP2 stable overexpression, there is higher mRNA level of ecathedrine. So in summary, cell lines from the invasive bladder cancer have an EMT gene expression profile, and the key inducer appears to be ZEP1. On the other hand, two non-invasive bladder cancer lines have a gene expression profile typical for epithelial cells, with high expression of ecathedrine. At the same time, in non-invasive lines, we may see a higher expression levels of ESRP1 and ESRP2, which may indicate their important role in the EMT process. Moreover, downregulation of ESRP1 and ESRP2 induces the decreased level of ecathedrine and simultaneously increases the level of vimentin and ZEP1. And finally, the stable overexpression of both regulatory proteins increases the level of ECAT in UMUC3 line. Learning more about the role as well as the exact mechanism of action of ESRP1 and ESRP2 in EMT process in bladder cancer lines will be a crucial part for a better understanding of the progression of this cancer. Thank you so much for your attention, and I'm happy to answer any questions now. Yes, and I would like to invite my fellow friends here on the stage, if I may.
As always, I have to ask, did you participate in any of the laboratory work or even watched it?
So we did most of the things which were done in the project. So starting from the beginning, we did all the RT-qPCRs that have been shown. My fellow friend did an RNA isolation. I was also trying to do the transcription from RNA to cDNA. Me and Veronica, we did Western blot, and I did the part of siRNA, so the transient knockdown, and the last part, so the stable overexpression, was done by Dr. Łukasz Sieroń due to our lack of time. However, I have talked to him, and I've known how he did it and all the steps which he had done.
Okay, I can see your understanding of this work. And did any of you participate in the statistical analysis, because there was plenty of statistical data?
Well, we did not, but however, I have accompanied the doctor which was doing it 2 times, frankly saying, and I was looking how she was doing it. Personally, right now, I wouldn't do it by myself, but I know how she was doing it, how she was creating it, so possibly in the future I will be also able to do it on my own.
Okay, thank you. Congratulations on
your
presentation and research.
Thank you very much.
I teraz przechodzimy już do pracy numer 8 i widzę, że jest też z nami autorka.
Good morning. My name is Zofia Kowalska and I represent the Department of Physiology at Medical University of Silesia. It's a pleasure to be here today to present our findings entitled Dynamic Changes in Interleukin 2 after subarachnoid hemorrhage, immune dysregulation and minocycline effects. Subarachnoid hemorrhage is life-threatening type of stroke that is caused by bleeding into a cerebrospinal fluid-filled subarachnoid space surrounding the brain. It accounts for approximately 15 to 20% of all strokes, the remaining majority being ischemic. Despite improved medical care, it still results in high mortality and disability. The pathogenesis behind SAH is multifaceted and it evolves over time with two different phases being recognized: EBI, early brain injury, and DBI, delayed brain injury. EBI refers to acute changes that occur within the first 72 hours after SAH and includes variable pathophysiological events such as elevated intracranial pressure, disturbed blood-brain barrier, global cerebral ischemia, and activation of neuroinflammatory pathways. DBI, on the other hand, it refers to a secondary complication that occur between 3rd and 14th day after SAH, such as delayed brain ischemia, cerebral vasospasm, and microthrombosis. The key force behind both EBI and DBI is immune dysregulation, characterized by complex reaction involving both central and peripheral immune system. This reaction is initially protective, but often becomes maladaptive, contributing to neuronal injury and poor clinical outcomes. Understanding the dynamics changes is crucial for developing targeted therapeutic strategies for improvement of recovery after SAH. The aim of the study was to monitor interleukin 2 levels over different time points after SAH to assess its role in neuroinflammation and to examine whether minocycline can stabilize its expression. Our study was performed on 120 male Wistar rats that were housed individually under controlled condition with free access to water and food. The rats were divided into sham, SAH, and SAH plus minocycline group with euthanasia 24, 72, and 7 days after. SAH was performed by injecting 250 microliters of non-heparinized arterial blood into the cisterna magna using stereotactic approach under anesthesia. The sham groups and the rats underwent catheter insertion only. Minocycline was administered intraperitoneally at 1 milligram per kilogram. Blood was collected, lymphocytes were cultured, and interleukin 2 level was measured using a bioplex multiplex immunoassay system. Results. So we observed that interleukin 2 level drops sharply at 24 hours and 72 hours after SAH, with a significant rebound at day 7 following the procedure. On this slide, we can observe minocycline groups alongside SAH groups, where minocycline appear to prevent the delayed spike in interleukin 2 levels after the procedure. Interleukin second is a key cytokine involved in immune regulation, best known for its dual roles in promoting activation of T cells and remaining and maintaining the tolerance through T regulatory cell. Our study suggests that interleukin second undergoes biphasic changes following the SAH. In acute phase, the level of interleukin second drops significantly, reflecting the immune suppression phase. How this decline may impair Treg survival, amplifying inflammatory cascade driven by neutrophils, macrophages, and activated microglia, all of which can contribute to further neuronal injury. However, by day 7, we observe that interleukin second levels rebound, potentially marking the shift between from immune suppression towards delayed compensatory activation phase. While it initially support tissue repair, unregulated late phase activation may contribute to neuronal injury and functional decline. Interestingly, minocycline, an anti-inflammatory agent, seems to stabilize interleukin second level in our study. While it did not prevent the early drop in interleukin second, it surely prevent the spike in interleukin second levels at day 7. This points to biphasic immune dysregulation after SAH, probably potentially correlating with EBI and DBI. In summary, interleukin second can serve both as biomarker and therapeutic target after SAH, and minocycline modulation of interleukin second can offer us hope for immune-targeted therapies after subarachnoid hemorrhage. However, further studies need to be performed to elucidate underlying mechanism and to confirm these results. Thank you for your attention.
A ja standardowo zachęcam do zadawania pytań i udziału w dyskusji.
I would like you to ask, because you all studied, I suppose, minocycline. What was your contribution? Did you participate in the laboratory work or even watched it?
Unfortunately, no, because I joined the research team recently, because last year. So I only did research on multiple interleukins, but I choose this one and only interleukin second, and I look forward to participate in further investigation about it, because it's only beginning on our work.
Okay, and another question. I don't know if it's on our side, but on most slides there were signs that it was generated by artificial intelligence. Is it on our side, or you used artificial intelligence to analyze the findings?
No, I did. I only use it to generate the graphics, cause.
Okay, but you analyzed it on yourself, by yourself.
Yes, I read a lot of articles, but the statistic analyses were performed by our tutor. So
okay, thank you.
I w takim razie myślę, że możemy przejść do kolejnej pracy.
Okay, so good morning, dear jury and fellow students. My name is Maciej Nowakowski, and today, as a representative of Department of Physiology, Faculty of Medical Sciences in Katowice, I would like to present you our research titled Changes in Lipid Profile After Sleeve Gastrectomy with Postoperative Monacolin K Supplementation. By way of introduction, we included this slide to explain keywords for our research, and that is obviously obesity and sleeve gastrectomy. And obesity is a major global health problem strongly associated with cardiovascular disease, type 2 diabetes, and, for example, dyslipidemia. But we should add to this description one more term, and that is visceral obesity, characterized by accumulation of adipose tissue, especially in abdominal cavity in greater omentum. And this type of obesity is mainly responsible for these comorbid conditions. On the other hand, sleeve gastrectomy is widely used bariatric surgery worldwide, and this procedure involves surgical removal of approximately 75 to 80% of stomach volume, and by this significantly reducing stomach volume, sleeve gastrectomy limits the amount of food patient can consume at one time, leading to earlier satiety and obviously to a lower overall calorie intake. We should also pay attention to monacolin K, that is a principal compound of interest in our study. And monacolin K is a natural substance found in red yeast rice. And that is polyketide, the product of fermentation of Monascus purpureus and three more yeast species. And its structure is nearly identical to lovastatin. Lovastatin, that is a common statin drug, cholesterol-lowering drug. And that's why we can use it to inhibit HMG coenzyme A reductase. Here we can see this enzyme. That is a main enzyme in cholesterol synthesis pathway. And the aim of our study was to evaluate the effect of monacolin K supplementation on lipid profile after sleeve gastrectomy, and to compare changes in total cholesterol, LDL, HDL, and triglycerides between supplemented and non-supplemented patients. Processing further to materials and methods, we can say that was a retrospective study. There were 149 female patients who underwent sleeve gastrectomy within the COSBAR program. What is important, all patients met the inclusion criteria of this COSBAR program, and here we can see this criteria. And 3 weeks after surgery, a subset of patients received monacolin K supplementation, while the rest formed control group. And the most important thing for our research is this point. Lipid profile parameters were measured at first before surgery, and then 3 months and 6 months after surgery. Moving into the results, at the beginning, we can look at the total cholesterol, and here we can see in experimental group the statistically important decrease of a level of total cholesterol. However, in control group, there is also decrease, but when we look at T0 and T6 and refer it to experimental group, this decrease is minimal. When we observe LDL measurements, there is obviously statistical important decrease of LDL in experimental group. And what is interesting, there is statistically important increase in control group between T0, that is this measurement before surgery, and T6, measurement after surgery. So this indicates that monacolin K can have a protective effect on LDL levels in patients after a sleeve gastrectomy. Going further to HDL, there is a statistical significant increase in both group, but the more important is in control group. And that suggests us that sleeve gastrectomy has alone a beneficial effect on lipid parameters on patients after sleeve gastrectomy, regardless of any supplementation. And last but not least, triglycerides. There is a significant decrease in both groups. However, the more significant is in experimental group, but in this case, we can say that can be a possible synergistic impact of a sleeve gastrectomy and monacolin K in measurement of triglycerides and the change of its level. And taking everything into consideration, a sleeve gastrectomy improves lipid profiles within 6 months post-operatively. Monacolin K supplementation enhances LDL and triglyceride levels. Monacolin K may be a valuable adjunct to post-operative care, but what is important? Further randomized studies are needed for confirmation and to evaluate the long-term safety and efficiency. Thank you very much for your attention. And obviously, I invite you to discussion. Okay.
Dziękujemy bardzo i
Okay, congratulations on your work. I would like to know, because I don't know if I got it right, when did you start supplement the monacolin?
After 3 weeks.
After the surgery?
After the sleeve gastrectomy surgery.
And did you measure the lipid profile right after the gastrectomy? Because
No. The first measurement was before a sleeve gastrectomy.
Okay, the
and
second
next
one
was 3 months after surgery.
So right at the beginning of the supplementation, yes? Am I?
No.
Am I right?
The monokalinka after 3 weeks, and we measured the lipid profile
Before starting the supplementation? Before starting the supplementation, did you measure the lipid profile before starting the supplementation and after the surgery? I'm talking about this 3-week period.
There weren't any measurements right before this supplementation after 3 weeks and right after this supplementation. The next measurement was after 3 months post-operatively.
Okay, thank you. I think now I get it. But what about this 3-week? I'm trying to get to the point to check if this 3 weeks period before this, if this period before start of the supplementation and right after bariatric surgery, how did the bariatric surgery implement the profile on its own?
And what? I don't know if you get it now, Mike.
We found some studies about own sleeve gastrectomy effects on lipid profile, and it is some of this, and it can be good suggest and good idea for the next study, for example, that we can make some study when we can measure after 3 weeks before start the supplementation of monocolin K.
To extract the exact effect of
But we have the control group that we didn't supplement the monocolin K, so our research was divided into the group who supplemented monocolin K and didn't supplement monocolin K. So we have the comparison to these two groups.
Okay, thank you. But I think that it would be also helpful to add to measure it on this particular timeline in both groups.
Yes, obviously. And that is, great idea how Victoria said for next research. But we should remember that was retrospective study where we collect data from hospital in Jaworzno, and that was only time when they use this supplement.
I get that. This was obviously the limitation of this study because it was a retrospective one. But from your point of view, if you had to conduct such a research, because it was a really interesting research in the future, so that's good that you acknowledge it and recognize.
Yes, and that's really good idea to measure in next studies these parameters right after bariatric surgery and then after these 3 weeks when we use this monocolin K. But in this research there was these two supplementation points, time points.
Yes, thank you for your explanation.
Thank you for this question.
Pytań ze strony tutaj online'owej, no to w takim razie dziękuję bardzo i zapraszam kolejnych przedstawicieli tutaj, w sensie przedstawicielkę.
Dzień dobry, Szanowni Państwo. Nazywam się Weronika Wójtowicz, jestem studentką 5. roku kierunku lekarskiego Śląskiego Uniwersytetu Medycznego w Katowicach. W imieniu naszego zespołu mam dzisiaj przyjemność Państwu przedstawić jeden z pierwszych wyników naszego projektu pod tytułem Ocena wpływu hiperbarii i hiperoksji na przejście epitelialno-mezenchymalne, a także oporność na leczenie temozolomidem w komórkach glejaka. Projekt ten jest realizowany w ramach grantu z projektu Studenckie Koła Naukowe tworzą innowacje. Glejak wielopostaciowy jest to jeden z najbardziej agresywnych nowotworów ośrodkowego układu nerwowego. Pomimo prób wdrożenia wielu nowych strategii terapeutycznych, żadna z nich nie wpłynęła na znaczący wzrost mediany przeżycia u tych pacjentów, która wynosi tylko średnio od 15 do 18 miesięcy. Standardową terapią w glejaku jest temozolomid w połączeniu z radioterapią, jednak z uwagi na to, że glejak jest nowotworem silnie inwazyjnym, a także jego wznowe, jest to tylko tak naprawdę terapia paliatywna. Wiele badań wskazuje na to, że mikroglej oraz makrofagi związane z glejakiem są zaangażowane w proces przerutowania, a głównym mechanizmem, który wydaje się być za to odpowiedzialny, jest przejście epitelialno-mezenchymalne, czyli EMT. Jest to proces, w którym zmienia się polaryzacja komórek. Co więcej, Zmienia się też ekspresja białek cytoszkieletu, a także macierzy zewnątrzkomórkowej. Głównym czynnikiem indukującym IMT jest czynnik TGF-beta. Wpływa on na wzrost ekspresji czynników transkrypcyjnych takich jak ZEP1, co skutkuje spadkiem ekspresji markerów epitelialnych takich jak ekadheryna i wzrostem ekspresji markerów mezenchymalnych takich jak wimentyna. Dodatkowo niektóre badania dowodzą, że IMT jest indukowane w warunkach hipoksji, a co więcej jest łączone z występowaniem lekooporności w komórkach nowotworowych. Z uwagi na silnie hipoksyczny charakter glejaka proponuje się zastosowanie nowych strategii, które wpłynęłyby na zmiany jego mikrośrodowiska, hamując białko HIF-1-alfa, a to spowodowałoby wzrost utlenowania komórek. Jedną z takich strategii wydaje się być tlenoterapia hiperbaryczna wykorzystująca tlen w stężeniu od 97 do 100% pod zwiększonym ciśnieniem. Z uwagi na doniesienia, że wpływa ona na wzrost wrażliwości komórek na radioterapię, jest proponowana jako nowa potencjalna terapia adiuwantowa w glejaku. Jednak potrzebne są jeszcze dalsze badania. Z kolei też inne doniesienia sugerują, że w warunkach hiperoksji IMT może być hamowane, a nawet może być indukowany proces do niego odwrotny, czyli przejście mezenchymalno-epitelialne. We wcześniejszych badaniach przez nas przeprowadzonych, które czekają na opublikowanie, wykazaliśmy, że w komórkach glejaka linii HROG17, która ma charakter wznowy, nastąpił spadek proliferacji komórek i spadek ich migracji, co zostało potwierdzone w teście gojenia ran. W związku z powyższym celem naszego projektu jest chęć ustalenia, jaki efekt ma tlen hiperbaryczny i także temozolomid na ekspresję genów związanych z IMT w komórkach glejaka linii HROG17 będącej wznową. W naszym projekcie wykorzystaliśmy pierwotną linię guza, mającą charakter wznowy, pozyskaną od pacjenta, scharakteryzowaną genetycznie. W eksperymencie komórki były podzielone na 4 grupy. Pierwsza grupa kontrolna, druga grupa po ekspozycji na temozolomid, trzecia po ekspozycji na tlen hiperbaryczny i czwarta po ekspozycji na temozolomid, a także tlen hiperbaryczny. Wszystkie komórki były hodowane w warunkach hipoksji, stężenie tlenu wynosiło 2,5%. Komórki, które były poddane tutaj działaniu tlenu hiperbarycznego, były umieszczone w komorze hiperbarycznej na 3 sesje, każda trwała 1 godzinę. Stężenie tlenu wynosiło od 97 do 98%, a ciśnienie 1,3 atmosfery. Następnie całkowity RNA został wyizolowany, przypisany na cDNA. Ten został poddany analizie z wykorzystaniem metody RT-PCR. Badanie ekspresji czynników transkrypcyjnych w komórkach glejaka linii HRG17 będącej wznową wykazało wzrost, wykazał nadekspresję czynnika ZEP1 w tych komórkach, co sugeruje, że jest to główny induktor IMT w tym typie komórek. Analizując ekspresję markerów IMT, wykazaliśmy, że zarówno w grupie po ekspozycji na temozolomid, jak i po ekspozycji na tlen hiperbaryczny nastąpił wzrost ekspresji ekadheryny, a był on największy w grupie, która była poddana ekspozycji zarówno na temozolomid, jak i tlen hiperbaryczny. Zauważyliśmy także nieznaczny spadek ekspresji wimentyny i ZEP1, który był najsilniej czy bardziej zauważalnie wyrażony w grupie po ekspozycji na temozolomid, a także po ekspozycji na temozolomid i tlen hiperbaryczny. Podsumowując, pierwsze z wyników naszego projektu wykazały, że zarówno tlen hiperbaryczny, jak i temozolomid powodują wzrost ekspresji ekadheryny w komórkach linii HROG17 mającej charakter wznowy, a ekadheryna jest czynnikiem, który hamuje IMT. Efekt ten, czyli największy wzrost ekspresji ekadheryny, jest widoczny w grupie, w której zastosowano zarówno temozolomid, jak i tlen hiperbaryczny. To sugeruje, że tlenoterapia hiperbaryczna może być nową potencjalną terapią adiuwantową w glejaku, jednak potrzebne są dalsze badania, również przeprowadzone na pozostałych liniach komórkowych glejaka. Planujemy je wykonać jako dalsze etapy naszego projektu, a także poszerzyć te badania o wykorzystanie metody Western blot, a także testu rysy. Dziękuję serdecznie za uwagę i zapraszam do dyskusji. Też zapraszam na scenę Karolinę.
Congratulations on your research. I'm impressed that you included your further research in your presentation. That's always good to know when you acknowledge that you studied only this part, but the further research is needed also on the other cell types. I would like to know, did you participate or watch any of the laboratory work?
Yes, me and Zuzanna Mielczarek isolated RNA. Carolina transcribed RNA to cDNA, and then me and Carolina performed RT-PCR, and then Carolina watched how Dr. Karolina Bajda-Krusinek analyzed the results.
Okay, thank you for the answer.
Thank you so much.
Dobrze. Przechodzimy do następnej prezentacji. Czy jest może na sali z nami tutaj Dominika? Super. No to tak.
Okay. Ladies and gentlemen, it's my pleasure to welcome you to the topic of effects of exercise after 8-day starvation on the fatty acid profile, a metabolomic analysis conducted at the Department of Physiology of the Medical University of Silesia in Katowice. We run on glucose, right? Aerobic respiration is based on glucose. But in the circumstances of prolonged fasting, the glycogen stores are rapidly exhausted in order to maintain the basic minimum concentration of glucose in blood. But we know that it's not necessarily the glucose itself that is needed to conduct the citric acid cycle. It's more acetyl-CoA that enters it. And that is how our body functions during fasting, obtaining this intermediate acetyl-CoA from different sources, such as ketone bodies, for example, 3-hydroxybutyrate, proteins, branch-chain amino acids to be precise, and lipids, fatty acids specifically. Fatty acids stored in white adipose tissue undergo beta oxidation, giving the acetyl residues that partially enter the path of ketogenesis and partially go to the citric acid cycle through elevated acylcarnitines that facilitate lipid transport into mitochondria. What's been done here during the study is combining those metabolic changes with exercise training. So the simultaneous synergistic effect between exercise and fasting has been widely investigated in the studies, and it has been proved that, of course, fat state exercise enhances performance, fat state meaning on a normal diet, but fasted state exercise, on the other hand, induces some metabolic adaptations in peripheral tissues. For example, fasted state exercise improves intramucellular lipid breakdown, leading to higher fat oxidation and greater fat loss. Exercise induces expression of skeletal muscle fat oxidative genes, and that process is actually blunted by glucose, so blunted by food. Purine and pyrimidine metabolism, anabolism is also increased during the fasted state exercise. And of course, different numerous metabolic pathways are altered. Now we stand in front of a question that maybe exercise is more metabolically beneficial during starvation? Well, we wanted to verify that. Aim of the study was to evaluate changes in the profile of selected plasma fatty acids following starvation and exercise. The study involved 5 healthy volunteers with no contraindications to prolonged fasting. The intervention included 8-day water fast, single bout of exercise, blood sampling, and mass spectrometry analysis. Prior to the study, all participants were on a mixed diet with small amount of meat for 3 days. Then the blood was collected under laboratory conditions. All participants took part in a progressive spiroergometric exercise test until the maximum oxygen consumption was reached, until the maximum exercise capacity was reached. And then blood was collected again 3 and 60 minutes after the end of the exercise. Then all participants began a period of 8-day fasting. It was a water fast, so they were allowed to consume only arbitrary amounts of the same medium mineralized water. Then the procedure was repeated, performed under the conditions of first rest and then exercise. Non-targeted metabolomic analysis detected changes in 243 metabolites, among which the following were statistically significant. The concentration of lauric acid was higher in the fasted state group than the fed state group when measured 60 minutes after the exercise. Referring to literature, lauric acid and high intake is associated with lower risk of type 2 diabetes. And research on mice has shown that it can reduce neuroinflammation caused by high-fat, high-fructose diet, and this basically happens probably through fatty acid translocase. Concentration of glyceric acid, on the other hand, was lower in the fasted state group than the fed state group when measured 60 minutes after the exercise. According to literature, glyceric acid, when accumulated at high levels, has been shown to be toxic, but on the other hand, some studies show that implementing arbitrary amounts of glyceric acid can be beneficial, for example, for liver or muscle function, and even reduce low-grade inflammatory factors such as interleukin-6 or glycosylated acute phase proteins. Concentration of pentadecanoic acid was first decreased 3 minutes after exercise and then increased 60 minutes after, but this change was statistically significant only in the fasted state group. Pentadecanoic acid represents a group of saturated odd chain fatty acids, and they in general are known for its beneficial properties. They're associated with lower risk of type 2 diabetes, heart disease, muffled, and even cancer. What's interesting is that isopentadecanoic acid, which is and basically an isomer of the previous one, behaved differently because there was a steady rise after the exercise, but also only statistically significant in the fasted state group. Moving on to the conclusions, I would like to stress that this is only a model, and such a long fast, 8-day fast, is neither enjoyed nor recommended. We have other ways of calorie restriction, such as intermittent fasting or time-restricted fasting, which can be implemented in everyday life. This model and 8 days were specifically designed to induce some significant metabolic changes and give some results, show the directions where to go. Then the exercise was a variable that was added in order to boost those metabolic changes. We believe that metabolomic approach to fasting may reveal its unexpected and maybe previously underappreciated effects, which may be later applied in, for example, diagnostic or therapeutic process. Thank you very much. I would be happy to answer your questions.
Thank you for your interesting presentation. I would like to know, because 8-day starvation seems like pretty radical way. Did you obtain any formal consent from the patient, as well the consent of the bioethical commission?
Yes, of course, the study got a consent of Research Ethics Committee, and however radical this may sound, the participants tolerated it well. They had a, let's say, personal doctor checking up on them regularly. So yeah, they underwent the fast well. During the study, frankly, there were some participants who decided to break the protocol because they couldn't stand it, but it was.
And you didn't include them
Yeah,
in
they
the
were
research.
included because the initial group was 8 participants. Then 2 of them admitted to not obeying the protocol, so they were expelled from the study, from the group. And there was even one participant who did not admit to breaking the fast, but it was possible to detect because her 3-hydroxybutyrate concentrations were too low to be true. So yeah, they could find it later on. Yeah.
Okay, thank you. And another thing that I want to know, do you plan, what type of control group do you plan to include in the future study? Because you said that was a model. What about future study? Do you plan to include any type of control group?
You mean a bigger group than this, or
No,
any
control group?
Because all participants were like a control group to themselves, because they were first doing the protocol, the exercise test on a normal diet. So
okay. Sorry, that was the point.
To eliminate the individual changes, it was done like this, that every participant did the test before the fast and after the fast.
Okay, thank you.
That the
I had
metabolism, yeah.
Okay, thank you. And do you plan to broaden the research to
yes, of course, because
bigger group of
yes,
patients?
the database is huge because there are, just as I said, more than 200 metabolites. So they're still being investigated. We are still looking for correlations. This is just a small piece of what the study is about, because I focused here on fatty acids because they seemed interesting. But we also have proteins, carbohydrates, and ketone bodies, which for sure are interesting when it comes to, for example, neuroprotective properties. And yeah, just as intermittent fasting is a popular diet, ketogenic diet is also popular, so maybe going into the direction of ketone bodies is not a bad idea also. Yeah, and the project is still on, so we're still working on it.
Okay, so we consider it as a pilot study, yeah?
Yes, let's say.
You. Thank you
okay,
for your
thank you very much.
Dobrze. No to teraz przechodzimy do następnej pracy, która jak widzę jest online. Czy jest z nami może przedstawicielka? Tutaj Kinga.
Yes. Oj, przepraszam.
Super, dobrze. No to poprosiłabym o udostępnienie jeszcze swojego ekranu z prezentacją.
Okay.
Dobrze, ja teraz jeszcze...
Okay, so can I start?
Sekundeczkę. Dobrze, możemy już zaczynać.
Okay, thank you very much. So, hello, my name is Kinga Wachowicz, and I would like you to present our study about the regulated post-vaccination status in the context of future maternity. So, have you ever considered why some pathogens are thought to be dangerous for pregnant women? So the TORCH complex is a group of pathogens that are especially dangerous for pregnant women. The acronym TORCH stands for toxoplasmosis, other agents such as Treponema pallidum or HIV, rubella, cytomegalovirus of herpes. And our study was mostly focused on the rubella virus. Rubella is said to be a mild infection caused by the rubella virus. The symptoms that it causes, it's such as rash or enlargement of lymph nodes, or for example, a mild fever. It's said that it mostly manifests in children between the age of 5 and 9. And scientists say that the 50% of adults that weren't immune to rubella and had contact with it will manifest asymptomatically. But although its disease is called a mild infection, the infection during the first trimester of pregnancy carries the greatest risks. Virus is transmitted to fetus through placenta, but it's also crucial to remember that the child may get infected by passing through the birth canal. Scientists say that infection in the first 12 weeks of pregnancy causes a 30% of chances of a stillbirth or miscarriage. And if the fetus survive, it can develop a lifelong disabilities or severe birth defects, also known as a congenital rubella syndrome. Congenital rubella syndrome can be also characterized as Greg triad. It is a visual defect such as congenital cataract, glaucoma, or retinoblastoma, some heart defects like patent ductal atheriosis, or deafness, which is a result of the damage to the auditory nerve. It is also known as the most common persistent sign of the congenital rubella syndrome. The way to prevent this infection from happening is to gain the immunity, so through the vaccination. Vaccine to the rubella is a live attenuated vaccine conducted with measles and mumps, also known as the MMR vaccine. It's highly effective because the first dose is set to give almost 100% of effectiveness after 2 weeks from the reception. And as I said, it's a live attenuated vaccine, so vaccination during pregnancy is contraindicated due to the low but still risk of teratogenicity. In 2023, Poland had the highest number because almost 76% of rubella cases reported in Europe. They were reported based only on symptoms, not on the laboratory diagnosis. So this is one of the reasons why this number may be so high. And World Health Organization also suggests that this is due to the fact of the increase of the anti-vaccination movement that started in Poland a few years ago. The aim of our study was to evaluate the level of specific anti-rubella antibodies among women of reproductive age. We studied a group of 30 women aged from 20 to 25, asked them a few questions about their current vaccination status. All of them answered that they were vaccinated according to the vaccination plan in Poland, which is the first dose of vaccine at the age of 1 and the second at the age of 6. We took their serum and made an enzyme-linked immunosorbent assay kit, ELISA. The walls of the microplate were coated with the antigen, to which we added serum with, if present, the antibody to rubella of our interest. Then we added another antibody, which was conjugated with an enzyme. And lastly, we add a substrate to this enzyme to cause a colorful reaction. In some wells, we added a known amount of antibodies to create the standard curve. And all of the results were measured using the spectrophotometer. The concentration below 8 was considered as a negative concentration, between 8 and 11 as a borderline, and above 11 or 11 as a positive. All of the women were positive, which meant that they were and are immune to the rubella virus. Total average antibody titer was 101.2. The lowest one was 22.9, but it's also above the 11, so this woman is still positive, which means that she still is has this immunity to rubella virus, and the highest one was 192.7. In conclusion, vaccination against rubella provides a long-lasting immunity. In order to maintain seroprotection, it is recommended to take another dose of vaccine if more than 15 years have passed since the last one. But the study group received their final dose of the vaccine at the age of 6, and despite the fact that it has been 15 years since the last dose, they are still immune to rubella. So as I said, vaccination against rubella provides long-lasting immunity. Thank you for your attention.
Dziękujemy bardzo za tą prezentację i tutaj również zachęcam do zadawania pytań lub też jakiejś dyskusji. Też chciałabym się zapytać, czy tutaj ze strony online'owej naszej jury jest wszystko w porządku?
Tak. Jakby pani mogła trochę opowiedzieć o wkładzie własnym pani w tą pracę.
Of course. So I did all of the research about rubella, about the congenital rubella syndrome. I also prepared all of the samples and made this ELISA test. And also I interpreted all of the results of the study.
Okay, thank you very much.
Thank you.
Dobrze, w takim razie, jeżeli nie widzę żadnych innych pytań, dziękuję bardzo i przechodzimy do następnej pracy. Czy jest może z nami na sali, bądź też, bo tutaj widzę, że stacjonarnie, ale czy jest z nami Martyna z pracy numer 13?
Tak, jestem. Miałam być stacjonarnie, ale
Dobrze.
szczerze mówiąc, dopiero mail o zmianie terminu mojej pracy ze środy na dzisiaj odebrałam, jak już byłam stacjonarnie w środę w Katowicach.
Oczywiście, nie ma żadnego problemu, rozumiemy. W takim razie czekamy na udostępnienie prezentacji. I jeżeli tylko wszystko będzie w porządku, to...
Czy już ją widać?
Tak, super. Dziękuję bardzo i jeżeli tylko będziesz gotowa, to możesz zaczynać.
Dziękuję. Ja nazywam się Martyna Szachniewicz i jestem studentką 4. roku kierunku lekarskiego na Kolegium Medicum w Bydgoszczy i działam w kole biologii komórki przy katedrze histologii. Przedmiotem naszych badań był wpływ flozyn na podstawowe czynności komórek raka pęcherza moczowego. Flozyny, czyli inhibitory kotransportera sodowo-glukozowego, są powszechne w praktyce klinicznej. Są one powszechnie używane w terapii cukrzycy typu 2, w przypadku niewydolności serca oraz również od sierpnia są one uznawane za ich działanie jest również uznawane za działanie nefroprotekcyjne. Więc skąd w takim razie pojawił się pomysł na to, żeby sprawdzić ich działanie na komórki raka pęcherza moczowego? A więc pierwszym uzasadnieniem może być to, że rak pęcherza moczowego występuje często u pacjentów obciążonych innymi chorobami, na przykład właśnie cukrzycą czy niewydolnością serca. W takim razie takim kuszącym wydawałoby się dostosowanie dla nich terapii, która oprócz leczenia podstawowej choroby przyniosłaby dla nich również inny pozytywny skutek na tą chorobę nowotworową, jak na przykład swoje zastosowanie znalazła rapamycyna w transplantologii. Oprócz działania immunosupresyjnego ona również wykazuje działanie antykancerogenne. Kolejnym takim czynnikiem, który mógłby nas popchnąć, który w sumie popchnął mnie do tego, żeby zastanowić się nad tym, czy to działanie może być wykazywane, były sprzeczne doniesienia literaturowe. To znaczy były udostępnione w literaturze opisy pacjentów, w przypadku których wystąpiła niemalże remisja po wprowadzeniu flozyn w przypadku raka wątrobowo-komórkowego, a w przypadku raka pęcherza moczowego, szczególnie w przypadku jednej z flozyn, których badaliśmy, czyli w przypadku dapagliflozyny, wystąpił wręcz progres choroby. I takim szczególnie dla mnie interesującym krokiem, który był taką pierwszą myślą, był mechanizm działania tych leków. Skoro one hamują kotransporter sodowo-glukozowy w kanaliku proksymalnym nerki, czyli zwiększają tym samym glukozurię, czy ich działaniem na przykład niepożądanym poza zwiększoną glukozurią i ryzykiem zoomu może być na przykład jakiś wpływ na komórki nowotworowe. I to było takim pierwszym krokiem. Nie jest to zawarte w tej prezentacji, ponieważ nie tyczy ona tego, ale pozwoliłam sobie to wyodrębnić jako taki punkt milowy w tych badaniach. A więc badanymi komórkami były komórki linii T24 raka pęcherza moczowego i wybrałam te komórki, ponieważ prowadziłam wcześniej hodowlę linii komórkowej różnych komórek raka pęcherza moczowego w innym laboratorium do pracy doktorskiej, kogoś. I szczerze mówiąc, jako że miałam ograniczony budżet swojej pracy tutaj z grantu studenckiego, to postanowiłam wybrać te komórki, bo były one dla mnie najbardziej dostępne i ich hodowla była dla mnie najbardziej korzystna. Bardzo dobrze się dzieliły i też są dobrze opisane w literaturze. Stosowaliśmy też dawki, które były dobrze opisane w literaturze na przykładach innych nowotworów i przeprowadziliśmy takie testy jak test MTT, zbadaliśmy apoptozę i nekrozę za pomocą aneksyny 5 i jodkupropidyny, zbadaliśmy cykl komórkowy, w jakim się znajdują te komórki, przeprowadziliśmy immunofluorescencję oraz test gojenia ran. Przejdźmy więc do wyników. No i możemy zobaczyć, że nasze wyniki już przy każdym z dobranych stężeń spowodowały istotnie statystycznie spadek żywotności tych komórek. Jednak największą tą istotność zaobserwowaliśmy w kanagliflozynie. Ale czy w takim razie ten spadek żywotności na ten spadek żywotności miała wpływ apoptoza albo nekroza? Okazuje się, że nie, ponieważ jak możemy zobaczyć tutaj na wykresach, nie miały one żadnej istotności statystycznej. Po 24 godzinach inkubacji z flozynami nie zmieniła się zarówno żywotność komórek żywych, ani tych apoptotycznych, czy ilość tych komórek nekrotycznych. Co to w takim razie znaczy? Znaczy to tyle, że musiał być więc inny mechanizm, który zmniejszył żywotność komórek. Proponowanym przeze mnie tym mechanizmem jest hamowanie również kotransportera SGLT2 w komórkach raka pęcherza moczowego. Ale żebyśmy się w ogóle dowiedzieli, czy ten kotransporter się tam znajduje, przeprowadziliśmy analizę Western blot. Niestety nie ma mi tutaj na prezentacji, ponieważ są to nasze świeże badania, ale ze wstępnych wyników jesteśmy w stanie stwierdzić obecność tego kotransportera na komórkach raka pęcherza moczowego linii T24. Przechodząc do analizy cyklu, zaobserwowaliśmy również, że z każdej z działań flozyn wiąże się spadek ilości komórek w fazie podziału oraz zablokowanie tych komórek w fazie G0 i G1. Chcąc sprawdzić również, jaki wpływ na migrację czy też podziały mają flozyny na komórki, przeprowadziliśmy barwienie fluorescencyjne. I tutaj możemy zobaczyć, że w dwie z dawek, 10 mikromoli oraz 20, możemy zauważyć, że również w każdej z tych flozyn nastąpił spadek ekspresji danego wimentyny czy F-aktyny. Najbardziej widocznym spadkiem zarówno wimentyny, jak i F-aktyny zaobserwowaliśmy w przypadku empagliflozyny. Jak już mówimy o migracji komórkowej, przeprowadziliśmy również test gojenia ran. I tutaj możemy zaobserwować, że najbardziej istotny wynik po 24 godzinach, 10% jedynie zmniejszony co do naszej kontroli, znaleźliśmy w kanagliflozynie. Ma się to również podobnie do tego, co zaobserwowaliśmy w teście żywotności. Przechodząc już do końca mojej prezentacji, chciałabym powiedzieć, jakie widzę dalsze kroki mojego działania. Jak już mówiłam, jesteśmy w trakcie analizy Western blot, czyli jest to pierwszy krok, którego dokonamy, ale takim moim planem, może poniekąd małym marzeniem jest to, żeby przeprowadzić również analizę immunohistochemiczną na próbkach pacjentów i zbadać tam ekspresję kotransportera SGLT2 oraz zobaczyć korelację z danymi nie tylko histopatologicznymi danego guza, ale również danymi klinicznymi. Co może być jedną przeszkodą jest to, że kotransportery barwią się, są problemy z ich barwieniem, ale już znalazłam dane literaturowe odnośnie raka wątrobokomórkowego, z którym ten transporter został zbadany, jego ekspresja została zbadana i myślę, że bazując na metodach, które tam w tej pracy zaimplementowano, będę mogła również swoje tkanki zabarwić. Dziękuję bardzo.
Dziękujemy bardzo za tą prezentację i jak po każdej zachęcam do zadawania pytań i udziału w dyskusji.
Ja mam do Pani takie pytanie.
czy były prowadzone badania z jakąś kontrolą? No bo tutaj jakby pani badała wyłącznie dose response na tych próbkach raka pęcherza. Czy próbowała pani również analizować, jak te flozyny wpływają na komórki zdrowe, na przykład na komórki nabłonkowe w warunkach in vitro lub fibroblasty w warunkach in vitro?
Dziękuję za to pytanie. Takim jeszcze jednym z kroków, który owszem muszę zamieścić, jednak nie będzie to w ramach tego badania, który chcę zrobić, jest właśnie badanie wpływu na komórki prawidłowe raka pęcherza moczowego, ponieważ owszem nie ma tutaj próby kontrolnej.
Dobra, dziękuję. No myślę, że warto to rozważyć, bo też stężenia flozn, którymi pani działała, wydają mi się dość duże. W sensie takim, że to mimo wszystko są dosyć wysokie stężenia w porównaniu z tymi leczniczymi na obwodzie uzyskiwanymi u pacjentów. Ale
Te stężenia były, dziękuję, te stężenia, jak już mówiłam, były dobierane na podstawie danych badań in vitro i innych liniii komórkowych dostępnych w literaturze, na przykład raka piersi.
Dobra, rozumiem. Ślicznie dziękuję.
Dziękuję bardzo.
Dobrze, widzę, że nie ma więcej pytań. W takim razie bardzo dziękuję za tą prezentację. I przechodzimy już do następnej, do prezentacji numer 14. Zapraszam.
Dziękuję bardzo. Szanowni Państwo, nazywam się Klara Ferenc i reprezentuję Koło Naukowe Katedry Biologii Molekularnej Wydziału Nauk Medycznych w Katowicach. Chciałabym dzisiaj przedstawić Państwu pracę, w której skupiliśmy się na modulowanej przez białka cytotoksyczności oraz interakcjach błonowych gwiaździstego polimeru kationowego DMAM. Słowem wstępu kationowe nanopolimery stanowią obiecujące syntetyczne wektory do dostarczania kwasów nukleinowych do komórek. Ich dodatni ładunek sprawia, że mogą się łatwo kompleksować z kwasami nukleinowymi, promują endocytozę, ale także wiążą się z ryzykiem powstawania tzw. korony białkowej, która może w znaczący sposób wpływać na ich właściwości fizykochemiczne oraz biologiczne. To z kolei może prowadzić do upośledzenia dostarczania do komórek, a także wiązać się z silnymi, niespecyficznymi interakcjami błonowymi z wysoką cytotoksycznością. Dlatego celem naszej pracy była analiza, jak białka surowicy mogą wpływać na cytotoksyczność oraz interakcje błonowe kationowego gwiaździstego polimeru DMAM, a także ocena formowania się i właściwości potencjalnie występujących kompleksów polimer-białko. W tym celu wykorzystaliśmy komórki linii HT1080, które inkubowane były z gwiaździstym polimerem DMAM w obecności lub bez 10% roztworu surowicy. Badania składały się z 3 głównych etapów. Najpierw była to analiza cytotoksyczności oraz ścieżek śmierci komórkowej, następnie analiza interakcji polimeru z błonami, natomiast ostatnim etapem była analiza tworzenia się kompleksów polimer-białko. Tak więc zaczęliśmy od analizy żywotności. Tutaj główną obserwacją było to, że sam polimer wykazywał bardzo wysokie efekty cytotoksyczne, natomiast w obecności białek cytotoksyczność malała. Co więcej, podczas barwienia oranżem akrydyny zauważyliśmy, że sam polimer prowadził do śmierci komórkowej, indukując procesy związane z nekrozą, natomiast w obecności białek surowicy dochodziło do śmierci komórkowej indukowanej w procesach apoptozy. Tutaj widzimy na rycinie komórki właśnie umierające śmiercią nekrotyczną w przypadku braku tej surowicy. W związku z tym założyliśmy, że polimer sam w sobie musi w jakiś sposób uszkadzać błonę. W związku z tym kolejnym etapem naszych badań było zbadanie przypuszczalności błony oraz potencjalnych jej uszkodzeń. W tym celu skorzystaliśmy z technik cytometrii przepływowej, korzystając z 2 popularnych barwników. FDA, który w przypadku uszkodzenia komórek jest wyrzucany z komórki, co prowadzi do spadku zielonej fluorescencji, a także jodku propidyny, który wnika do komórek i interkaluje z DNA jedynie w przypadku uszkodzenia ich błon. Nasze wyniki jednoznacznie wykazały, że wolny polimer zdecydowanie uszkadza błonę, co zaobserwowaliśmy jako wzrost populacji komórek, które wykazywały sygnały dla jodku propidyny oraz spadek ilości populacji komórek, które wykazywały sygnał dla FDA. Co więcej,
w obecności surowicy uszkodzenia błony malały, co jednocześnie tłumaczyło, dlaczego w przypadku braku surowicy dochodziło do śmierci nekrotycznej, a nie apoptotycznej. Ponadto sprawdziliśmy też przyczepność polimeru do komórek, łącząc go ze znacznikiem fluorescencyjnym. I tutaj z kolei wykazaliśmy, że w przypadku braku białek surowicy dochodziło do zwiększonej jego przyczepności. Obserwacje te potwierdziliśmy również mikroskopowo, korzystając z technik mikroskopii fluorescencyjnej i postawiliśmy hipotezę, że być może polimer wiąże się z białkami znajdującymi się na powierzchni błony. Żeby to sprawdzić, zastosowaliśmy trawienie trypsyną, jednak trawienie to nie spowodowało spadku fluorescencji, w związku z czym hipotezę tę musieliśmy odrzucić. Natomiast spadek fluorescencji wykazaliśmy przy potraktowaniu komórek tritonem, czyli detergentem, który uszkadza bezpośrednio błony komórkowe komórek. I przez to, że właśnie zaobserwowaliśmy całkowity spadek tej fluorescencji, założyliśmy, że w ciągu przynajmniej tej krótkiej inkubacji, ponieważ trwająca jedynie 5 minut, żaden z polimerów nie dostał się w znaczącym stopniu do wnętrza komórek. W związku z tym postanowiliśmy wydłużyć czasy inkubacji, co spowodowało, że faktycznie po traktowaniu tritonem wyniki były nieco inne, ponieważ fluorescencja nie spadła, co sugerowało, że doszło do endocytozy, co również widać tutaj czy może tutaj akurat nie widać, ale gdyby się bliżej przyjrzeć, byłyby widoczne małe czerwone punkty, które zwykle są związane właśnie z zajściem procesu endocytozy. Kolejnym punktem naszych badań była właśnie analiza tworzących się kompleksów polimeru z białkami. I w literaturze jest opisane takie zjawisko, które nazywa się spontanicznym formowaniem korony białkowej na powierzchni nanocząstek pod wpływem kontaktu z płynami organicznymi, głównie z surowicą. Korona białkowa jest strukturą bardzo silnie dynamiczną, która potrafi w znaczący sposób modyfikować właściwości biologiczne nanonośników, a ponieważ zauważyliśmy, że białka modulują w znaczący sposób zachowanie naszego polimeru, zastanawialiśmy się, czy w przypadku być może właśnie też białek surowicy i naszego polimeru 2MA nie dochodzi do powstawania właśnie takiej korony. I tutaj okazało się, że faktycznie po zmieszaniu polimeru z białkami dochodzi do powstawania większych kompleksów, które były mniej rozpuszczalne, a ponadto wykazywały one dynamikę czasową, co zdawało się być spójne z powstawaniem i z potencjalnym powstaniem korony białkowej. Następnie wykonaliśmy analizy na DLS-ie, które pokazały nam, że faktycznie powstają agregaty, które mają od ponad 100 nanometrów do ponad 200, które były także widoczne w transmisyjnej mikroskopii elektronowej. Dodatkowo potencjał zeta pokazał, że podczas połączenia polimeru z białkami surowicy jego ładunek stał się zdecydowanie mniej dodatni, co z kolei tłumaczyło, dlaczego polimery w obecności białek surowicy mniej chętnie przylegały do komórki. Tak więc tutaj podsumowując najważniejsze wnioski wynikające z przeprowadzonych badań, wykazaliśmy, że polimer 2MA wykazuje silne działanie cytotoksyczne w medium bez surowicy, indukując nekrozę poprzez zaburzenia błonowe. Po drugie, białka surowicy obniżały toksyczność polimeru, jego adhezję do komórek, uszkodzenia błonowe, a także przekierowywały komórki na szlaki śmierci apoptotycznej. Po trzecie, polimer błonowy 2MA łączył się raczej z fosfolipidami błony niż z białkami oraz prawdopodobnie mógł ulegać endocytozie. Po czwarte, polimer 2MA formował duże kompleksy z białkami. Jednak zasadniczym wnioskiem wynikającym z tych badań jest to, że białka surowicy znacząco obniżały cytotoksyczność tego polimeru poprzez ograniczenie jego interakcji błonowych oraz promowanie formowania się dużych, ujemniej naładowanych kompleksów. Badania te podkreślają znaczenie interakcji polimer-białko dla zachowania nanonośników, a także ich potencjalnych zdolności transportowych. Co więcej, badania te naszym zdaniem otwierają również pewną możliwość stworzenia pewnej otoczki białkowej wokół gwiaździstego polimeru 2MA, który mógłby się na przykład składać z rekombinowanych ligandów białkowych, które pozwoliłyby na interakcję z odpowiednimi receptorami na konkretnym typie komórek, na którym by nam zależało. I w związku z czym mogłoby to stanowić pewne nowe rozwiązanie dla terapii celowanych. I jest to też kierunek, który chcielibyśmy dalej rozwinąć, jeśli tylko uda się nam otrzymać odpowiednie finansowanie. Dziękuję bardzo serdecznie za uwagę i zapraszam do dyskusji.
Proszę coś powiedzieć, jest w ogóle bardzo ciekawa prezentacja. Jeżeli mogłaby Pani coś powiedzieć, w jaki sposób taki polimer można zastosować w leczeniu? W sensie to jakieś implanty są, czy to się po prostu podaje? W jaki sposób taki polimer można dalej zastosować?
Znaczy przede wszystkim tutaj chodzi o leczenie nowotworowe, przeciwnowotworowe. Możemy połączyć ten polimer na przykład z jakimiś cząsteczkami siRNA, które niszczyłyby ekspresję pewnych genów albo indukowały na przykład apoptozę. Natomiast jeśli chodzi o samo podawanie tego polimeru, dużo się mówi właśnie o terapiach celowanych, natomiast praktyka, przynajmniej na tyle, na ile jestem zorientowana, wykazuje, że jednak nie ma specjalnie póki co możliwości faktycznego zastosowania klinicznego takich rozwiązań. Dlatego też na razie skupiliśmy się po prostu na badaniach in vitro, testując dostępny polimer, który akurat został dla nas syntetyzowany w Centrum Materiałów Węglowych i Polimerowych w Zabrzu. Natomiast jeśli chodzi o jego dalsze zastosowanie kliniczne, bardzo byśmy chcieli, żeby to było możliwe. Natomiast póki jeszcze nie wiemy, czy jest on na tyle na przykład bezpieczny dla komórek zdrowych bądź nie, no i jeszcze nie zastanawialiśmy się tak bardzo nad pozostałymi aspektami dotyczącymi chociażby aplikacji późniejszej w leczeniu klinicznym tego nanopolimeru. Mam nadzieję, że odpowiedziałam na pytanie.
Tak, z mojej strony bardzo dziękuję.
Dziękuję bardzo.
I would like to congratulate you on this research. It is really well thought on.
Dziękuję bardzo.
As well, congratulate you on your style of presentation. The only one thing I would like to say to you is maybe for future presentations, I would advise you to stand to face the audience, because in here it's not a problem for us, but on a further conferences, it might be a problem, and that would be too bad to take points from you because of that. Because your research is really well thought of.
Bardzo dziękuję za uwagę. Od razu chciałam wytłumaczyć właśnie też na większości konferencji, na których wcześniej byłam, zwykle mównica była ustawiona właśnie przodem do widowni i widziałam podgląd, natomiast przez ilość wyników uznałam, że lepiej monitorować, o czym mówię, niż później dopuścić właśnie do jakichś nieporozumień, że tak powiem. Ale bardzo dziękuję za uwagę i będę pamiętać na przyszłość.
Thank you. I think that in that case it is even better to just to print out the results. It looks better. Just it looks better.
Yes,
Thank you.
Dziękuję bardzo za uwagi.
I teraz przechodzimy już z tego, co widzę, do naszej ostatniej prezentacji, jaką mamy dzisiaj zaplanowaną. Jest ona tutaj online. Czy jesteśmy na łączach z prezentacji?
Yes, hello.
Dobrze, super. Cieszę się, że się słyszymy. W takim razie proszę tutaj podpiąć kamerkę, załadować prezentację i jeżeli tylko uzyskamy, że tak powiem, pełną gotowość, to proszę informować. To zaczniemy.
Jeszcze tylko sekundę.
Oczywiście.
Hello, my name is Krystian Czołpiński and I'm associated with Center for Regenerative Medicine in the Medical University of Białystok.
Przepraszam, jeszcze tylko chciałam zapytać, bo tutaj się kamera jakby włączyła, ale nie widzę dalej obrazu, czy...
Czy teraz lepiej?
Super. Tak, dziękuję bardzo.
Zaczynając od początku. Hello, I'm Krystian Czołpiński and I'm affiliated with Center of Regenerative Medicine from the Medical University of Białystok. I'd like to start my presentation by telling you a little bit something about asthma. Asthma is a very chronic and heterogeneous disease affecting many people worldwide. It results in epithelial damage, chemokine induction, bronchial hyperactivity, goblet cell metaplasia, and airway remodeling, on which I want to focus. Airway remodeling is a complex interplay between collagens, MMPs which digest those collagens, and TIMs which inhibit the function of MMPs. Asthma can be generally split into three endotypes: high T2, low T2, and non-T2, of which low T2 and non-T2 are the least responsive to current treatments and thus result in higher inflammation and higher remodeling. Additionally, there are no clinical models relevant for them up to date that are well proven. So that's why we aimed to use novel house dust mite extract-induced model to quantify those changes in remodeling-related cases. The model looked like this. So we stimulated the mice for 5 days with 2 days break intranasally for the period of 2 weeks and 12 weeks, representative for acute and chronic respectively, using 10 and 100 micrograms for acute and 100 micrograms of HDM for chronic model. We started our analysis by looking at the histological structure of our lungs. So we looked at H and E stainings and have seen increased infiltration of inflammatory cells in all analyzed models. And we also looked at the PAS staining representative for mucus, shown in the reddish color. And we have seen its increased production in all analyzed models, with also a little bit of flexing in the bronchus in the chronic model. Next, we aimed to quantify the frequency of immune cells using flow cytometry, and we have seen the increased frequency of interleukin-4 plus cells coupled with increased frequency of interferon gamma cells and interleukin-17 cells in acute models, and the predominant increase of interleukin-17 plus frequency of several interleukin-17 plus cells in HD100 chronic model. We also observed increased frequency of interleukin-10 plus cells in acute models, which represent, and all these findings represent that acute models represent low T2 asthma, while the chronic ones represent non-T2 asthma. We also wanted to look at the collagen deposition to see the remodeling changes using Mason's tree from staining, and we have seen increased deposition in all analyzed models, although the collagen was a little bit covered by the increased inflammation in the chronic model. Next, we aimed to do curated analysis to see what genes were up or down regulated in these cases, and we have seen increased upregulation of collagen-related genes in acute models, while these changes were not so prevalent in the chronic ones. We also looked at the MMPs and TIMPs, and we have seen that the patterns were very similar, except for some of them, like MMP17, MMP8, TIMP2, MMP11, or TIMP4. So to look at the collagens in more depth, we performed immunohistochemistry stainings and quantified the collagen's deposition subepithelially for collagen 1A1 and 3A1 and total deposition of collagen 4A1. We observed increased deposition of collagen 1A1 in HDM100 acute model, while deposition of collagen 3A1 and 4A1 was increased exclusively in HDM100 acute and chronic models. To see the further interplay between collagens and MMPs, we quantified the MMP levels in bulk samples using Luminex analysis, and we have seen increased levels of MMP2 in HDM100 both acute and chronic models, and increased levels of MMP8 and MMP9 in HDM100 chronic model, and also MMP12 was increased in all analyzed models. To see the interplay with MMPs and TIMPs, which inhibit those MMPs, we performed also Western blot analysis, and we have seen a statistically significant decrease in TIMP1 level proteins for acute models, with a trend of decrease in all analyzed TIMPs except for TIMP4, in which we observed statistically significant increase in HD100 chronic model. So to see whether the dysregulation in collagens deposition and also gene expression was due to the MMPs and TIMPs interplay, or whether it was as a result of different cell death types, we performed tunnel assays, and we have seen increased apoptosis. What is surprising is that it was mainly in inflammatory cells in acute model, and what is even more surprising is that we didn't detect any apoptosis in chronic model. That's what prompted us to analyze other cell death types, like for example necroptosis, which involves RIPK1 and RIPK3 interplay, which are phosphorylated to their active forms. Then their active forms then phosphorylate MLKL, and this active MLKL oligomerizes in the cell membrane, which prompts to release dumps, which further the inflammation and thus result in higher array remodeling. So to look at this, we performed immunofluorescent analysis, immunofluorescent stainings of RIPK1 and its active form, showing a little bit of increased signal, but the signal for RIPK3 and its active form was more dominant. And what is the most interesting is that the signal for MLKL and its active form was really increased in the chronic model, prompting us, which is of course the last path in the cell death type, and it makes the channels in the membrane, and thus it prompted us to believe that necroptosis is mainly predominant in chronic model. So to sum everything up, we have seen that HDM induced mice model is good. It reflects the T2 low and non-T2 inflammation well, that this inflammation results in a higher array modeling related changes which we characterize using the interplay between collagens, MMPs, and TIMPs, and that we observed increased apoptosis in acute models, while necroptosis was mainly observed in chronic ones. I would like to thank everyone for their support in this work, and I will await your further questions.
I would like to know if you participated or watched any of the laboratory work.
Yes, of course. I was able to participate in the mouse models, and additionally, I participated in other essays, mostly in Western blot analysis of the samples.
Okay, thank you.
Dobrze, w takim razie, jeżeli nie ma więcej pytań, to dziękuję bardzo za tą prezentację.
Thank you very much for your time.
I to była również ostatnia prezentacja przewidziana na dzisiejszą sesję. Tego właśnie chciałabym poinformować, że wszystkie zgłoszone prace zostały zaprezentowane. Gratulujemy wszystkim autorom prezentacji oraz uczestnikom sesji. Chciałabym Wam szczególnie podziękować, dlatego że jak dobrze widzieliście, to sesja ta miała 2 części, co świadczy o tym, że rozwijamy się i jest tych prac coraz więcej. Mam nadzieję, że z roku na rok będzie ich jeszcze więcej i że będziemy mogli mieć już nawet 3. część. I chciałabym jeszcze kwestii formalnych i nieformalnych. Z kwestii formalnych chciałabym zaprosić przede wszystkim Was na ceremonię wręczenia nagród i ceremonię zakończenia konferencji SIMC, która odbędzie się na sali A3 dzisiaj o godzinie 17 oraz z takich mniej formalnych rzeczy oczywiście chciałam zaprosić na After SIMC DSO, które się zaczyna również dzisiaj o godzinie 21. Dziękuję Wam jeszcze raz za uczestnictwo. Te osoby, które gdzieś tam też były z nami online również dziękuję. Dziękuję komisji online, dziękuję komisji stacjonarnie i uważam konferencję, tą część tej sesji za zakończoną. Dziękuję.
Bardzo dziękujemy za możliwość tutaj udziału i za wysokie dla wszystkich uczestników brawa za naprawdę wysoki poziom prac naukowych.
Dziękujemy bardzo.